The brain, a wrinkly mass of cells that sits protected in the ironclad fortress known as your skull. A mass of electric activity that coordinates the vast complex network that is the body, all in the effort to preserve the continuity that is your DNA. And buried deep within the brain are two almond-shaped complexes called the amygdala. The center is partly responsible for the experience of fear. In an environment of safety, people have been known to seek out experiences that trigger a fear response. I'm not entirely sure why, on an evolutionary scale, why people do it, why they would voluntarily press on that amygdala and put themselves in a position to fear. But when I looked at my YouTube comments, I came to understand that my audience is no exception. Prion proteins have a special property by which pathogenic variants of the protein can cause other non-pathogenic prions that the body naturally makes to misfold and aggregate. These toxic protein aggregates slowly build up in the neurons, leading to neurodegeneration and death. What makes prions so terrifying is that their aggregates cannot be cleared naturally by the body and are basically impossible to degrade enzymatically. Typical cooking temperatures were also insufficient to decontaminate affected beef, meaning cows infected with mad cow disease, a type of prion disease that has the capability of traversing the species barrier to infect humans, had to be incinerated. This biological agent is slow-acting, deadly, ultra-stable, ultra-persistent, and because it is unlike any other pathogen, no therapeutics for it exist. Those properties would be sufficient for the prion to reign as the king of nightmares, but on top of all of that, because misfolded prions cause healthy prions to misfold, they have the potential to grow exponentially. Prions are some of the scariest pathogenic agents I have ever seen. Some of you even believe that prions make classically horrifying pathogens like rabies virus look like amateurs. So here it is. This is almost everything I have ever learned about prions. Before we learn about prions, let's get on the same page when it comes to terminology. A prion is any protein that, when misfolded, can cause other proteins of the same type to misfold. But for clarity and simplicity's sake, when I talk about the prion protein, I am specifically referring to the protein product of the major prion protein gene. There are many prion proteins, but the one that will be relevant for most of our discussion of mammalian diseases is this major prion protein. And before we talk about prion diseases, we should first talk about what the major prion protein actually does in its healthy form. And that's a surprisingly difficult question to answer, involving a scientific roller coaster of seemingly contradictory results. Typically, when biologists want to study the function of a protein, they'll take a model organism and genetically modify the organism in a way that renders that protein non-functional. This is called a genetic knockout, an animal whose gene of interest has been disabled. We can then compare the biology of the knockout animal to the wild-type animal, or the animal that has not been genetically modified, and deduce the function of that gene. The first engineering of a major prion protein knockout in mammals were the Xeric-1 mice, made in 1992. How did the researchers break the prion protein gene in the Xeric-1 mice? Before the advent of CRISPR, a common strategy to break genes was to do a domain swap. What this involves is identifying the gene sequence of the protein of interest and taking advantage of a property of DNA called recombination that allows scientists to replace a huge chunk of the gene with some other DNA sequence, breaking that gene. The Xeric-1 mice no longer have a functional major prion protein gene because the majority of the prion gene was replaced with a neomycin resistance gene, producing a Frankenstein protein that should prevent real prion protein from being made. The neomycin resistance gene codes for an enzyme that confers resistance to the antibiotic neomycin and has no real function in neurons, making them a good candidate to break the prion protein without too many side effects. The researchers subjected these engineered mice to a battery of behavioral tests to check their cognitive functions through a series of mazes. What was shocking was that up to 7 months in age, these mice were largely normal, with no significant differences in their ability to learn. It seemed that the Xeric-1 mice lacking major prion protein being normal could lead one to believe that healthy prion protein has no major biological function. But hold on, scientists are nothing if not thorough. There are some interesting questions we can still ask given the results of the Xeric-1 study. 1. The whole of the prion protein was not removed. Maybe the little bit that was still being made still has a function, and not removing the whole protein wasn't drastic enough to cause a change. 2. Maybe the function of the prion protein is a function of old age. 7 months of age is safely an adult mouse, but it isn't geriatric. Many prion diseases take some time for symptoms to show. 3. Navigation of mazes is certainly a great test for assessing the cognitive, behavioral, and motor ability of a mouse, but maybe the prion protein is important for a somewhat complex/subtle change not captured by these tests. 4. Even if we didn't learn much about the prion protein's native function, we can still ask an interesting question about prion diseases. Prion disease is hypothesized to be propagated by misfolded prions forcing other prions to misfold. Do mice lacking the prion protein exhibit resistance to prion disease? I won't go into too many details on these particular experiments, but the short conclusion to this question is yes, the xeric 1 mice lacking prions were resistant to infection by prions, supporting the hypothesis that prion diseases were in fact caused by prions. To address the first issue of the xeric 1 mice potentially making some weirdly functional version of prion protein, a different group created another engineered strain, this time preventing the mouse from making prion mRNA or prion protein altogether. This line was called Edinburgh and replicated the results from the xeric 1, showing that removing the prion protein did not have a measurable effect on developing mice. Not only was the Edinburgh mice less molecularly messy, the Edinburgh mouse also came from an inbred line, meaning there was less of a chance that the genetic engineering could have had unintended consequences. So that's it, right? Major prion protein has no major function in mammals. Knocking it out twice in 2 different mice lines showed no noticeable effects. Surely scientists would have moved on from this, but no. A third group made a mouse line called Nagasaki, which had the whole prion gene replaced with an antibiotic resistance gene. Their rationale for making this mouse was not apparent from their papers. Maybe they had trouble getting the existing mice or something, or maybe they took issue with how the previous mice were engineered. Whatever the case may be, unexpectedly, in contrast to xeric 1 and Edinburgh, the Nagasaki mice in their old age experienced difficulty walking and had a marked decrease in the number of Purkinje cells in their brain. Purkinje cells are special neurons found in the cerebellum, the cortex of the brain responsible for motor coordination. This presents us with some conflicting data. Xeric 1 and Edinburgh were largely normal. Nagasaki mice showed that prion protein might play a role in motor coordination. Why were the results different, and which one of these results should we believe? When the genetic engineering strategies were carefully compared, the Nagasaki mice were found to have removed a bit of extra DNA at the beginning of a protein coding region of the prion gene. This extra deletion was later shown to have major unintended consequences that resulted in the discovery of a secret, hidden gene lurking in the shadow of the original prion gene. I'm pausing here to allow you to pick up the pieces of your surely shattered brain. This gene was called Doppel, for being the prion gene's doppelganger, and it lives 16 kilobases downstream of the prion gene. Doppel is an especially appropriate name as its sequence identity is so similar to the prion genes that it is hypothesized that Doppel and prion shared the same genetic ancestral origin. Show of hands how many of you prion freaks out there knew there was a prion doppelganger gene. I sure as heck didn't. It turns out that the extra bit of deletion that the Nagasaki mice had removed is what is called a splice acceptor site. We all know that DNA codes for mRNA, which is then read to make protein. But it is less known that mRNA is often processed in eukaryotes before it is made into protein. Eukaryotic mRNA is made of introns and exons, and before protein is produced, the introns are spliced out. Different combinations of splicing introns out can result in variations in protein structure, giving one stretch of DNA the ability to code for many different proteins. The prion protein is made of 3 exons that are read to make the protein, and these 2 introns here have to be cut out of the mRNA or the protein will be made incorrectly. These intronic sequences are removed through a process called splicing. In the healthy normal mouse, the prion gene and the Doppel gene make the mRNA for 2 different proteins. But by removing a splice site at the top of the prion gene sequence, the resulting production of the mRNA gets messed up, making a very long mRNA that includes the Doppel gene. In other words, the Nagasaki mice not only broke the prion protein, but also resulted in the production of way too much Doppel. Later research has confirmed that the overproduction of Doppel and not the removal of the prion protein was responsible for the Nagasaki mice losing coordination in their old age. And I think that's the raddest fucking shit I've ever seen. Progression in prion research has now allowed us some insight as to what the true cellular function of the prion protein is. It just wouldn't make sense for evolution to have conserved this gene across so many walks of life, in spite of its potential for biophysical corruption. If it were truly useless and truly just harmful, evolution would have likely jettisoned this gene from the genome long ago. We now know, after improved methods of research, that the prion protein is a membrane protein that is primarily made by neurons. We also know that the prion protein is significantly enriched at the axon and the presynaptic terminal. The axon is the long body of the neuron and acts as a relay for electrical signals to travel through. Synapses are junctions between neurons that allow one neuron to pass off signals to another. One could hypothesize that the prion protein is important for the structure and/or function of the synapse. While we discussed genetic knockouts as a way to investigate the role of genes, scientists can do the opposite. Overexpression lines are genetically engineered mice that make too much of a protein of interest. Mice that make too much healthy prion protein have increased synaptic transmission, meaning that the connections between neurons get stronger the more prion protein you have. But before you start taking prion supplements, just know that stronger neuronal connections don't always result in improved performance. Biology is about being just right for your context, meaning there is such a thing as neurons being too strongly connected. A hypothesized function of the prion protein is in the regulation of sleep. This certainly makes sense, given that prion disease fatal familial insomnia is a disease in which patients lose the ability to sleep. However, a direct link between prions and sleep has been difficult to prove and may be good to cover in a Phy bit. Prions might also play a role in mitigating neurotoxic damage. Ironically enough, mice without prion proteins have larger stroke volumes than mice that have prions. It's kind of weird. Finally, prions are hypothesized to play a role in myelin maintenance. Myelin sheaths are wraps of proteins and lipids that surround the body of the neuron to increase conduction speed of brain signals. Myelin sheath degradation is associated with all sorts of neurodegenerative diseases. To conclude this chapter, we can say that prions may play a subtle but important role in neurons. We might not need the protein to survive, but I think we're pretty sure we wouldn't be the same without making that protein. It's still a mystery why the prion protein has had such strong evolutionary pressure to be maintained in mammals, and I'm excited to hear what scientists have to say about it in the future. We now know a bit more about what prions actually do for us. Now we'll finally get into why you clicked on this video. Prion diseases. But since I'm not a medical doctor, nor do I want to inflict you with a neuroses about your health, I won't be talking about the diseases themselves, but the molecular biology behind how the diseases can manifest. Quick recap. Misfolded prions cause properly folded prions to misfold. Misfolded prions clump together in aggregates that the cell is not equipped to break up. Eventually, the garbage gets out of control and the cell dies. How does a good prion misfold and turn bad? The details are somewhat fuzzy, but one way is that genetic mutations cause the amino acid sequence within proteins to change, and that slight structural change may make them far more vulnerable to being misfolded. We know that one end of the prion, called the N-terminus, is very disordered, meaning that part of the protein has a very high level of floppiness. The other end of the protein, called the C-terminus, is very structured, ordered, and relatively inflexible. Several mutations in the C-terminal end of the protein have been associated with the development of prion diseases, and those specific amino acids have been shown to be important for keeping those parts of the protein together. If any of these amino acid sequences change, say by spontaneous somatic mutation, the generally high-ordered C-terminal end of the protein can fall apart, allowing for messy, aggregation-prone configurations. This protein, whose sequence is now mutated, can now interact with healthy prion protein, forcing those proteins to misfold as well. If all prion diseases are caused by misfolding prions, why do these diseases have different symptoms? FFI starts with insomnia, CJD starts with dementia, Kuru starts with changes in motor ability. I might go into more depth in a Phy bit, but the short end of it is it's pretty mysterious, and might have to do with where those prions are aggregating. Why do cells die when dealing with prions? At least one mechanism by which prion aggregation causes cell death is by stressing the organelle known as the endoplasmic reticulum. Not only is the endoplasmic reticulum the hub for protein production in the cell, it's also the site in which these proteins are folded with the aid of proteins called chaperones. This quality control function of the ER is really important. It's estimated that 10% of healthy prion protein is initially made improperly folded. So the ER's job of correcting prion structure before it can be transported and poison other healthy prion proteins is as you can imagine, of paramount importance. It is hypothesized that a buildup of misfolded prion protein in the ER, potentially induced by contact with an infectious prion, causes the ER to go into hyperdrive, upregulating the unfolded protein response. This program aims to hold onto misfolded proteins, unfold them, refold them, and send them on their merry way. Since misfolded prion proteins are so hard to unfold and refold, these toxic prions build up in the ER. Chronic activation of the unfolded protein response activates the p38 MAPK protein, initiating the cell death pathway known as apoptosis. A reasonable assumption to make is that if we can figure out how to unfold and misfolded prion protein, or how to destroy them in a biologically relevant way, that a future in which these diseases are treatable is at the very least imaginable. So let's take a look at what I found about the structural biology of the prion protein. We know that misfolded prions are nigh impossible to destroy in a biological context, but why haven't we figured out a way to destroy them reliably? One reason why research into this problem is so tough is due to the inherent difficulties of studying the aggregated protein form. We've actually known the crystal structure of the healthy prion protein since 1996. Knowing what proteins actually look like on a molecular level is an incredible weapon for scientists to engineer solutions to these problems. We could, for instance, design something that maybe binds the healthy prion protein and prevents it from binding a diseased prion protein. But that would require understanding the origami steps by which contact with a misfolded prion causes a healthy prion to misfold, and that's difficult because it's really hard to find out what a misfolded prion even looks like. See, X-ray crystallography has been an incredible tool for molecular biology, the pinnacle of structural information. But its power comes with a drawback. As the name implies, you need your protein to form an actual crystal. As we learned in part 3 of this monster of a video, prions containing those specific amino acid changes have more disorder than their healthy counterparts, meaning getting them to form something like a crystal is really difficult. All hope is not lost for prion structural analysis. We just have to go a bit more low-res. Technologies like FTIR and CD spectroscopy can tell us a lot about the features of prion aggregates, namely that they are characterized by a shift from alpha helices to beta sheets. A paper using a technique called cryo-EM, which is an incredible technique with resolving power almost as good as X-ray crystallography without the drawbacks, was able to see the ultrastructural elements of a specific type of prion aggregate called a fibril, with proteins of their full length. Many prion structure studies had to cut off bits and pieces of the protein that were not amenable to crystallization. But this is the first I've seen where the whole-length proteins giving rise to fibril aggregates were captured, and I gotta say, they are incredible. You can actually play with the models online, link in the description. Prion fibrils appear to involve stacks of these beta sheet heavy structures that have all of this surface area for interaction. Now, I'm not a structural biologist, but I hypothesize that one reason why these aggregates are so hard to break down with heat is that stacks and stacks of intermolecular forces combined makes molecules ultra-stable. While understanding prion protein structure may hold the key to developing treatments for prion diseases, I was personally unable to find therapeutic strategies that took advantage of this new knowledge. Maybe in the future, scientists will develop a targeted molecule that can disrupt these interactions, but for now, my focus shifts elsewhere into Currently, to my knowledge, there are no approved treatments for prion disease. There were many promising drug candidates in the past, but many trials have failed in the animal model stage. One such drug is quinacrine, a drug that started to work in mice, decreasing the amount of prion aggregates, but then failed as those prions had adapted a drug-resistant arrangement. My goodness, that is a scary sequence of words. Drug-resistant prion disease. I suppose since there aren't any drugs even out right now for prion diseases that it's not inherently worse, but still. Another cool attempt at a drug is the anti-prion AP1. The anti-prion is a prion whose aggregates do not appear to cause disease in mice. The idea here is that antiprions form their own aggregates that are resistant to disease-related folding patterns, effectively preventing disease-related prions from spreading their poison. When treated with AP1, mice still get accumulation of prion aggregates in their brain, but their brains are surprisingly normal, and they were resistant to developing clinical symptoms of prion disease. Insanely cool, but I haven't really seen a clinical trial come out of that technology. There is one promising ongoing clinical trial for an anti-prion drug, maybe the first ever. Ion 717 is an experimental drug that is an antisense oligonucleotide. What this basically means is that the drug is a piece of mRNA that binds prion mRNA. When those mRNA molecules bind to each other, they are destroyed, lowering the amount of prion protein that can be made in the brain that can be misfolded. This option could allow those who have prion aggregates building up in their brain to delay the progression of their disease. As we've seen early in the video, the prion protein does appear to have a function, but it doesn't seem to be required for life. In medicine, sometimes you have to make trade-offs. When faced with something as harrowing as a prion disease, side effects may be an acceptable risk to take. This was the perfect time to make this prion video, as the clinical trial started in 2024 and is predicted to end in 2025. If the folks at Ionis Medicine can pull this off, it would be an incredible victory for humanity. Let's hope that they do. While the folks at Ionis Medicine are in the middle of trials, it's not good to put all eggs in one basket. There is still research into basic prion biology, chemistry, and physics that are still going on. Perhaps we need to go deeper, more basic. Nope, too basic. Yeast are known to make different prion proteins all the time. Given they're not mammalian major prion protein, but they are proteins that do prion things. Evolution happens so much faster on the scale of microbes, and yet these organisms are still making a ton of different prion proteins. Why? In their non-aggregated form, many of these prion proteins have a healthy function. When they aggregate, it's actually really bad for the yeast. There is one notable exception, and it's crazy. It's the Het-S system. The Het-S aggregated prion, the form of a prion that is most associated with dysfunction, may actually help the yeast exchange DNA properly. Basically, if two yeast strains get a little funky and want to swap DNA, if they're of the same strain, then all good, go ahead. However, if one fungi makes a Het-S form that can form prion aggregates and the other doesn't, when the cells merge, the merged cell dies. When Het-S prion interacts with Het-S soluble, the Het-S soluble protein changes shape into a protein that destabilizes the cell membrane. Why might a fungi want that? These fungi often grow as colonies, a conglomerate of cells that cooperate with one another. An improper fusion between two incompatible cells may result in a functional enough but unhealthy cell. By having a system by which improper fusion events can be detected and removed, these fungi ensure a high level of evolutionary fitness in their environment. Functional prions. That's so cool. It makes sense, following the fact that fungi are prolific prion producers, that they evolved anti-prion defense systems, including chaperone proteins that help prions unfold and refold properly. Maybe a future therapy will take inspiration from the humble yeast. Another microorganism that surprisingly deals with prions are freaking lichens. Lichens are assemblages of fungi and algae. Out of seemingly nowhere, lichens have enzymes that are in fact capable of degrading prion aggregates. I had no idea there were any enzymes that could degrade prions, but the humble lichen apparently makes proteases that break down a wide range of prion variants, some down to 1/100th of their initial concentration. And that is an absolutely incredible thing to learn. One of the most absolutely horrifying scenarios you can think of is slowly accumulating prion aggregates in our food chain. But as far as I know, that hasn't happened yet. And why not? It's possible that microbes, like lichens, have evolved to break down the prion protein, preventing this apocalyptic scenario. But because the authors of this lichen prion review paper are cautious, I will echo their caution. I haven't read any proof about lichens being remediators of diseased prions in the environment, but boy, that would be awesome, wouldn't it? Prions have always been a dark fascination of mine, commonly thought to be one of the most terrifying biological agents to exist. But I didn't want to focus solely on what makes prions scary, because that covers the spotlight on all the incredible work that all the scientists that I've cited have done. If you like this video, subscribe for more deep dives into molecular biology. Thank you for watching. This video's fan art spotlight goes to icosahedron. If you want to show off your art, please use the hashtag #microbialmasterpieces and you might get a spot in the next video. If you want to support me beyond subscribing, why not sign up for my YouTube membership like these lovely people here, where you'll get access to the private-ish Discord and hang out and talk biology and stuff, as well as get sneak peeks of whatever it is I'm working on at the moment. See you next time.